everyone, I'm Katie, a PhD candidate at McGill University, and welcome to Translating Cancer Research. In this podcast series, we will be discussing ways to more rapidly and effectively implement fundamental research findings into the clinic to improve the lives of patients with cancer. We will host scientists, clinicians, industry professionals, and patient advocates to help us paint a picture of the translational research process and determine how we can optimize it. Before we get started, though, I would like to thank our two sponsors. Our first sponsor is the McGill Centre for Translational Research in Cancer, under which we have founded a student committee that is conceptualizing, hosting, and editing all parts of the podcast. We will talk a bit more about the MCTRC in the upcoming episode with Dr. Batist, but just to give you a brief intro, the goal of this center is to provide a solid foundation based on state-of-the-art platforms, multidisciplinary training, and academic industrial partnerships for the advancement of scientific knowledge, specifically at this interface between fundamental and clinical research. Secondly, I would like to thank the Tremplin Consortium. Selected from a competitive pool, our podcast was granted the Tremplin Ambassador Award, which helped us acquire all our recording equipment and software that we need for this show. The consortium reunites trainees across all McGill-affiliated research institutes with the goal of empowering biomedical trainees, strengthening community ties, and promoting a strong professional development infrastructure in the life sciences sector. This includes various funding opportunities, workshops, training programs, and networking events. For both the MCTRC and the Tremplin Consortium, if you'd like to learn more about them, you can check the description where I've linked their websites and LinkedIn pages. And now for the episode. So with us today to introduce both the podcast and the topic of translational research is Dr. Gerald Batist. He is a clinician scientist, the director of both the Siegel Cancer Center as well as the McGill Centre for Translational Research in Cancer. Throughout his career, he has made significant contributions to cancer research and the development of novel clinical trials. So welcome, Dr. Batist, and thank you for joining us today.
Thank you. It's great to be here.
So I wanted to start off the episode by talking about translational research itself. I was hoping you could give us your definition of translational research, keeping in mind that some of our listeners might have never heard of the term.
Yeah, it's interesting. It's a term that first appeared in the United States scientific report, presidential report, and I think in the early '90s, which is what sort of stimulated us thinking about creating such a center. And the idea is taking something from the laboratory to the clinic. So fundamental research, how do we make this into something that we could at least test for the benefit of patients? Translational research, that's in its crudest terms. And since then it's expanded in its definition, and we understand it in a much more clear way. And that is, it's not just a point of transition between the lab and the clinic, but it's actually a whole domain. So there's translational research that goes from fundamental discovery into very specific kinds of animal models, where you may have transgenics or you may have patient-derived xenografts, and you can test a lot of things there. And that's a whole transition from lab to kind of a preclinical state. And then there's the other end of the domain, which is taking the things you learn there into the clinical setting and testing them. And that's another interface that is less well developed, I would say. And so translational research is the whole gamut of all of these things.
Okay, interesting. Yeah, you were talking about these different interfaces. So generally, basic research can potentially move into a translational type setting where we create these preclinical models. We try to test drugs in this setting, but it's really moving this preclinical research into the clinic. That's where a lot of research doesn't move, and that's one of the major difficulties.
Yeah, that's one of that domain. The second one, moving from all these preclinical models, which have taken years to develop, into clinical testing and doing the early phase trials, that's what our MCTRC is really focused on, generating the clinical trials. And then there's another component, which is what do we learn from the early phase clinical trials that we can take back to the lab in order to refine things and then take it back to the clinic. So there's an iterative process there. But that's that end of that domain of translational research is where our focus is.
Okay. Yeah, so like you're saying, it's actually more of a circular process. It's not just from one end to another, but, even phase 3 clinical trials can help us inform basic research.
Yes, as a matter of fact, it can. For example, me and Connie Sopsey, a PhD graduate, just finished publishing a paper where we actually hooked onto a clinical trial, a phase 3 clinical trial that was being done by a large pharma, got hold of the samples and tried to define if we could find biomarkers that would predict which patients were going to respond and not, using the endpoints of the clinical trial. We discovered a whole new set of biomarkers using a phase 3 clinical trial, and that then it turns out to be a metabolomics pathway, and that then has to go back to the laboratory to be refined and discovered. And in order to even measure those things, you have to develop assays for some proteomic assays. And so, yes, even clinical research, clinical specimens can take you right back to discovery research, can be very rich in information. A few years ago, we did a study called the Winther study, where we biopsied patients' tumors, metastatic tumors, and then we analyzed not only the genomic profile, but the transcriptomic profile, the RNA. And we both discovered that extended the number of targets or different therapeutic options for those patients, but also some very sort of fundamental discovery that even if you had, say, for example, an amplified gene, doesn't necessarily mean that you have more transcript. Even if you have what we in the laboratory call a activating mutation, it should be turning on the gene, you don't necessarily have more transcript. So there are disconnects at each level, and that, I think, required us going back to the laboratory to unravel it. So it's a very dynamic process.
Absolutely. So I was hoping before we jump into more of the research that you're doing, that we could talk about the McGill Centre for Translational Research in Cancer. Right. And this is something that I think you developed back in the 90s at some point. So could you tell us a bit about what it is and what its purpose is?
Yeah, so in fact, this was in response to the development of this essentially neologism, this kind of new word, translational research, that was created in this presidential scientific report. And we decided, well, it would be important to actually try to create something like this at McGill and take advantage of the very strong fundamental research and clinical research and find a path between them. And so we were developing a network anyhow, and the idea was to appeal to people that were at that interface or on either side, but very interested in the other side of the interface, and bring everyone together. And over the years, it's changed a few times. We've brought it through McGill University Senate. It's a formally approved center, and with membership both inside McGill and outside McGill. It's based at the Lady Davis at the Jewish General Hospital, but has tentacles really throughout Quebec. And so the idea is to capitalize on the knowledge that we all have, so that we can share, and particularly here to be focused on that interface that we just discussed, taking some of the fundamental discoveries that people have and seeing how we can fashion them into something that can be tested, either as a biomarker that we can validate or as a drug that we can try, etc. So over the years it's developed. We were among the first to start biobanking in the early 90s, and that's evolved dramatically. And we also started an experimental pathology laboratory that that is very, very high quality, and we see them popping up in a variety of other centers as well. But we provide core services for people to be able to make this interface.
Yeah. Well, I actually, what I wanted to ask was, at the beginning of the MCTRC, before it was developed, how was clinical research? What were the interactions like between fundamental researchers and clinicians at that time? And I guess it could have been a limitation to bring things forward and to improve treatment.
Yeah, there were. It's an interesting question. There were. It's a great question. There were interfaces between clinicians and fundamental scientists, but they weren't smooth, and they were not iterative. So a lot of people in the laboratories were interacting with clinicians, mostly surgeons, just to get clinical samples so that they could study them. Or occasionally someone would bring some tissue or tissue from a patient that had failed a therapy in order for us to study in the laboratory why they might be resistant to that therapy. But the goal actually was to turn around and not only figure out why they're resistant, but then figure out ways of overcoming resistance, maybe develop a treatment, and then go back to the clinic and see if we could actually improve the therapy. So there always were some interactions, and I think for many years, some of us fooled ourselves to think that there were fundamental scientists who were just interested in knowledge and really didn't anywhere in their heart want to see an application of their discovery. But in fact, I think everybody who's working in the most fundamental of research really would like to see some implications for human beings. And so that's the space that we're trying to fill.
I think that's definitely true for a lot of students as well that are getting involved in translational research at this point. I mean, it's really nice to see that there could be a potential impact of what you're doing in the lab. And so I think that actually motivates a lot of students to get involved in translational research. And that's what the MCTRC is really good at supporting for these students.
Yeah, I think that it's kind of a fulfilling a natural kind of need to, even though as a student you're really focused on the head of a pin, one particular molecule or one process, it's nice to feel that it fits into something that ultimately has impact. And I think that's a very satisfying feeling. And I think it also provides some pathways for a career, because in fact, sometimes, in order to move forward in translational research, you're necessarily interacting with commercial entities, either with startups in technology, biotechnology, or in pharmaceuticals. And again, that's an interface of the MCTRC as well. We're interacting with companies and commercial entities so that we can test new technologies and introduce new drugs into their pipelines. And that's, I think, something for students to learn as well, because some of the career paths are not academic. Some of them are into that world of pharma or biotech, and I think having some kind of look at that during your training is interesting as well.
Yeah, I think so too. And that I guess it's kind of interesting because a lot of us are studying, like you said, we're focused on a very, very specific niche, I guess. And so oftentimes if we do discover something, I know in my case I have a drug combination that I find really interesting, and I want to understand its potential implications and if it could have an indication in the clinic. And I realized that if I really wanted to make this happen, ultimately it's me, the driving force behind it, and I might need to really be the one to push it to go further and further. And
it's exciting to think that and know that you can. That you can do that. And I think that's very, very important for everyone to understand, and particularly for the students.
Mm-hmm. And I think one of the major goals of this podcast is to really understand how we can do that,
Right.
And what are the biggest limitations to doing that. And so yeah, it's motivating to be in this type of research, but trying to figure out how to move from one end of the spectrum to another, or one end of the translational research spectrum to another continuum.
Yeah.
Trying to figure out how to do that is quite complicated, and that's the point of this podcast. So so I wanted to ask you, since just back to the MCTRC again.
Yes.
Since its genesis back in the 90s,
Mm-hmm.
how have you seen the translatability of research improve?
Well, first of all, our technologies advance very, very quickly. So, just not even doing it ourselves anymore, just access to archived databases of genomic information, transcriptomic information, and now here with Christoph Borchers, we're and CP Tech, we're looking at proteins, the ultimate target of drugs, and so there's much more information about targets, and it's very easy to get this information about specific patients. Liquid biopsies we may talk about as well, which don't require a biopsy. And then at the same time, pharma has been exploding with new targeted agents. So the combination of these two things has moved everything forward very, very rapidly. As we start to identify, the whole concept of precision medicine is that a cancer isn't a uniform diagnosis, that a breast cancer actually is a collection of rare and uncommon subgroups. And once we start to be able to define the subgroups, each one of them has their own natural history, and then each one of them has their own molecular drivers that we can try to inhibit. And then the mechanisms of resistance are not uniform either. And so we can start to develop, identify them, and develop molecules to try to overcome them. So it's been greatly accelerated by advances in technology and pharma buying into this. You understand, for pharma to have bought into precision medicine was a challenge for them, because once, for example, in the case of Herceptin or HER2 targeting agents in breast cancer, once we understood that was only maybe 15% of breast cancers, they weren't going to be selling this to everyone. It lowered their market. They had to just come to terms with it. But that one drug made a big difference for those women with breast cancer. Now it's re-expanding out as we start to understand that HER2 can be a target used to deliver other payloads. But so everything has advanced, both from the pharma side and the technologic side. Our ability to analyze tissue. We don't necessarily have to maintain tissue banks, which have cost hundreds of millions of dollars, to freeze tissue, and tissue that may be degrading in ways that we don't even know over the years. Now we understand that just fixed tissue that's very standard from the pathology lab can be used for all kinds of analyses, including even phosphoproteins. So I think the technology and the drug development, all of this has come together at a dramatic time.
Mm-hmm. And I think even regarding MCTRC, the placement of just having it sort of squished between the hospital, or the McGill Cancer Centre and the hospital, being in such close proximity to patients and being able to collect data and have these collaborations between fundamental researchers and clinicians, I think it's critical, and it must really help and sort of ease the research that you're doing.
Yeah, I think physical proximity can't be underestimated. It's overestimated. It's very, very important. So we built, for example, in the first iteration of the McGill Cancer Centre, the clinical research unit, where we do a lot of phase 1 studies and collect a lot of biosamples to take back to the lab, is in the heart of it. was right opposite the clinic, and now the treatment center, and now it's moved to an expanded area, but still just one floor down. So it's very important to have close proximity. That attracts another key element to translational research, which is clinician scientists. Not everyone can do that. Not everyone is that. That's not everyone's aspiration, but there are people who have both clinical knowledge and clinical practice and run research labs, so they understand the questions and the feasibility of asking and answering the questions in the clinical setting. And those kinds of people are busy both in the clinic and in the lab, and having the physical proximity is extremely important.
Mm-hmm. Like you, I guess.
Like me, yeah. I'm an example of a clinician scientist. It's a very rewarding and interesting career, but it's very challenging as well to maintain funding and at the same time protect your time and at the same time be in the clinic.
Mm-hmm.
But you have the opportunity to actually move things to the clinic and run clinical trials and be involved in clinical trials so that you understand the feasibility of testing something that's come up in the lab.
Yeah, I can imagine it's quite helpful to have expertise both in the lab and obviously
right,
as an
but
oncologist.
you don't act. But everyone doesn't have to be that. But these people can be linkers and can help connect PhD scientists with MDs who have very little knowledge of science. So you have kind of these bridges that are extremely important to the whole mix.
Because we can't all be an expert in everything. So that communication is critical. That collaboration is critical.
Gone is the days, yeah. Exactly.
Yeah, especially with the amount of data we have now.
Right.
Okay, so for the next part of the podcast, I wanted to move on and talk a bit about the research that you do in the lab and clinical trials that you're involved in. So I thought we could first start talking about your research lab. What is it that you do in your lab? What do you focus on? And what are the sort of techniques that you're using?
Yeah, thank you. So I have been all of my career working on mechanisms of resistance to a variety of antineoplastic agents. And that gives you the other way of looking at that is mechanisms of sensitizing tumors to treatments. We, for a long time, worked on redox chemistry, and some of the drugs we're using and radiation generate oxygen radicals, and that directly takes you to systems that control that, like glutathione-related enzymes and glutathione itself. We did the first study of radiation and a drug called BSO, which inhibits the synthesis of glutathione with biopsies, and we were able to show that we could deplete it much more rapidly in tumors than in normal tissues. The whole issue here is distinguishing normal tissue from tumor. You don't want to sensitize normal tissue to potentially toxic treatments. So over the years, a variety of mechanisms of resistance have been discovered. We've been part of some of those discoveries. And then ways of blocking those mechanisms of resistance have also come up. And it turns out that when tumors become resistant to one drug, they often become resistant to a variety of drugs. We call that pleiotropic resistance. And so inhibiting one doesn't really do the job. You have to be able to inhibit all of them. And so slowly but surely we've worked our way upstream to a master transcription factor called NRF2, which turns on a whole cytoprotective battery of genes, including drug efflux, drug metabolism, redox related defense mechanisms, etc. And when NRF2 goes up, cells are resistant to a variety of treatments. And it turns out that there's a protein that controls NRF2. It's called KEAP1. And when KEAP1 is mutated, as it is in about 10% of some kinds of cancers, NRF2 is constitutively elevated, and those tumors are resistant. So if you can decrease the NRF2, you can sensitize the tumor. Problem is, you don't want to just decrease NRF2 in normal tissue and And tumor, so we are currently working on a molecular glue, these molecules that fix the mutation in KEAP1, bring down NRF2, and sensitize only the tumors that have the mutations to a variety of treatments. And we've been working for about 5 years now with significant funding, and we now have some lead compounds that do exactly that, even in animal models. So now we're at the point of talking to pharma about licensing this and studying this in the clinic.
Okay, so where did this NRF2 lead come from? Is it from, you said biopsies from patient samples?
Yeah, it came from biopsies, talking to colleagues, and just studying actually even in cell lines, starting to understand the mechanisms. We had a lot of different sort of downstream mechanisms of resistance, but once we understood that we needed to have an impact on multiple mechanisms, we started working upstream, and we found a common regulator, and that's
great.
NRF2. Yeah,
that's the goal.
Yes,
it does happen.
Right.
That's cool. Maybe I'll have to look back at my data because I saw NRF2 floating around.
Is that right?
A little bit. Yeah.
Well, that's great. NRF2 has implications in lots of things, and so does KEAP1, which regulates NRF2. But it turns out that it also binds PD1.
Oh.
And so it's going to have implications, perhaps.
That's a plus.
In immune checkpoint inhibitor. So in clinical studies, patients who have KEAP1 mutations do worse even with immunotherapy. So it has implications of resistance to a variety of kinds of treatment.
Mm. So
I have also over the years studied other things that we took directly into the clinic. For example, I helped with the development of liposome carriers for drugs, even classic chemotherapy, and we took liposomal doxorubicin all the way to the clinic, and it's approved and used in many countries.
That's great.
So you can actually see things that you work on help patients.
Yeah, I can imagine it's important to, while you're doing this basic research, to make sure that these protein targets that you choose, that they will in fact be able to at least select a set of patients that would possibly respond to a drug against them, as well as not having toxicity side of. So it must be really difficult to narrow down on a gene, on a protein, and to know that this could be a biomarker of a certain disease or a resistance. Right.
It's a process, but anything that you learn has some application in some way or another. It's just a question of once you have a very neat story, you can start with the whole story and then discover the mechanism, or you can discover a mechanism and then look for where it might be of relevance. Cancer is a very complicated disease, and involves multiple distortions of multiple pathways, receptors, etc. So whatever it is you're working on, if it's truly, if it's true, and you see it in biology, it must, even in the lab, it must have some implication somewhere in the clinic.
Mm-hmm. It's just about selecting the right set of patients to test it on so that it will get approved.
Right. And that's why, again, it's very important to be connected to the clinical world, to people in the clinical world, because they may recognize that what you're doing here has implication for a whole other type of cancer. Mm-hmm. And now we're in a state where we call it tumor agnostic drugs, tumor agnostic biomarkers, where the tissue of origin really isn't important, where you might see the same molecular profile in a subgroup of stomach cancers that looks like a subgroup of breast cancers. And so you need to have that clinical input.
Yeah, and that's also where precision medicine really comes in as well.
It has all kinds of implications, but for example, with the discovery of HER2 overexpressed subgroup of patients with breast cancer, about 15%, and the discovery of an an interfering antibody called Herceptin that changed the prognosis for those women dramatically. And because it was convenient for the pharmacists, we would often group them all together so that they were making all the Herceptin for that day and not wasting any. It turned out to be sort of a breast cancer clinic until it was discovered that a small percentage of patients with gastric cancer, which is predominant in men, also are HER2 driven. And so then we had a clinic that had men and women, different diseases, and it changed from being a breast cancer clinic to being a signature-based clinic, a molecular profile-based clinic. And that's happened and will continue to happen as we move away from anatomic diagnosis to molecular diagnosis.
Oh, that's really, really interesting.
Yeah, yeah, and it changes the culture of the clinic and who's in the waiting room and the entire context of the treatment.
Right.
Now we start to understand, for example, that you don't need a HER2-driven tumor to use HER2 as a target. So if HER2 is abnormally elevated, but just slightly elevated, it's not driving the tumor. It's not an amplified gene that you have to inhibit with Herceptin, but it is present, so you can hitch some kind of toxic cargo, either a radioisotope or a poison, to this antibody. It'll deliver it to the tumor that has even a slight overexpression of HER2, and you'll get a clinical effect as well. And so once we've kind of shrunk down and targeted a population, now we're starting to re-expand the population as we find other ways of using that knowledge.
Yeah, it's interesting because we have trastuzumab or Herceptin, which can bind targets and kill these cells. But like you said, it can also be used as a way to deliver, to recognize cancer cells. Even like you said, these as we're learning now about HER2 low breast cancer.
Exactly.
So these are breast cancers that express just a very small amount of HER2. Like you said, they're not driving the cancer, but they're there. And it turns out that we can use these HER2 targeting agents. We can indicate them for patients that before would have never received Herceptin because it's not the driving force behind that cancer. But now we're seeing that, like you said, we can actually expand and treat more patients with breast cancer with these agents that are delivering other drugs
and
to
with
these
this
tumors.
understanding, we've moved the other sort of up and coming area of therapeutics is theranostics. We're moving not only poisons but radioisotopes that have very narrow, short emissions, alpha particles in particular, but there are others. And so they don't do any damage to surrounding tissue, but you can connect those as well to trastuzumab or other targeting molecules, peptides or antibodies, and get the same kind of cytotoxicity. So it's a constantly expanding, both looking at the targeting agent, finding new targets, and the sort of toxic agent.
Mm-hmm. That takes us into the next part of the talk, which will be about, or the podcast. I know that you're working on some clinical trials, and you've had some obviously developed from your lab, but I know that you're also collaborating with other researchers and clinicians and pharma in order to start clinical trials that are ongoing right now. Would you be able to talk to us about a couple?
Yeah, I'll tell you about a couple, and sometimes it's serendipity how they connect. We're working with colleagues in Minnesota, and it's an interesting story. It began in 2017 or 2018 with a patient who had metastatic pancreas cancer, and she was doing really well, but right off the bat. But I started hearing about a colleague in Minnesota that was working with something novel. He had discovered that the bacteria Salmonella has an affinity. It's not a real formal binding, but an affinity for pancreatic tumors and some other tumors. So he deactivated it so that it's not toxic. It's still absorbed through the GI tract. He attached an immunostimulant molecule to that, and he could give that to patients, and they had no toxicity. It didn't in and of itself have any effect, but when he took domestic pets with spontaneous cancers, added chemotherapy to this Salmonella IL-2 combo, the dogs had dramatic responses and lived much, much longer. So he was at that point when I called him, and he said, yeah, we'd be interested in collaborating. So I called Health Canada, and I said, we'd like to do this as on a compassionate release basis. And the guy said to me, well, no, you can't do that. If it was a clinical trial, if he was already doing classical pancreas cancer chemotherapy plus this Salmonella molecule, you could do that, but it's not been started there. So I said, well, how about doing an N of 1 study? This is now something, again, in precision medicine that we talk about, where a patient has a unique molecular feature that fits with a drug that you can predict might be really beneficial to the patient. An N of 1 study, a single patient study. And the guy said to me, well, let me talk to my colleague. And in the end, they let us treat this patient as an N of 1 patient, and she did remarkably well. She ended up living 4 or 5 years, and I was able to taper off almost all of the chemotherapy. And we profiled her immune system, and her memory T cells were activated. Her cytotoxic T cell population was in the circulation was increased. So then we did a phase 2 trial. The survival of patients with metastatic pancreas cancer getting standard therapy is so standard and so consistent internationally that we didn't do a randomized study. We compared them to the same 25 patients treated here, same doctor, same pharmacist, same nurses the year before. And we treated actually 26 patients with FOLFIRINOX plus Saltikva, which is the name of this molecule. And in the end, we discovered that we had actually doubled the survival of the patients, doubled the time to progression of these patients. This was presented at ASCO last year, and then was in the Lancet Oncology list of top 10 presentations from ASCO, things to look at. So the manuscript is submitted and in review. But it's a major impact, we think, in a world in the pancreatic cancer world that has really not moved very much. So that's one example, and it really was a serendipitous connection between myself and the scientists in Minnesota.
I guess with something like pancreatic cancer, which is usually diagnosed at a late stage and often doesn't have a great prognosis, when you have an option for a drug or a treatment, a therapy, would you go right to Health Canada and convince them that this is necessary because it's the only option? Like, how can you bypass clinical trials with an N of 1?
Yeah, you're getting to a subject that is still a very live subject. How do we accelerate this next step? The FDA is very classical, asking us to do a randomized clinical trial. We're coming up with different clinical trial designs because it requires raising an awful lot of money. But for example, the design that we're settling on, and we'll see if this is acceptable to regulators, is if we treat a whole cohort of patients but do all of the immune profiling and we find that the benefits, the patients who have evidence of immunostimulation are the ones that live longer, that's much more convincing than doing a randomized study that may take a few years. There's the issue of delaying treatment. This is lives lost, months lost, time lost. The longer we wait, to get things into clinical practice, the more significant the losses to patients. And more drugs in development are really quite effective. 20 years ago, we weren't used to seeing in phase 1 studies actual tumor responses and quite dramatic responses. So as we're getting smarter in drug development, there's a lot of really good scientists in pharma now. We have to move all of this forward. So it's a very active and live debate. How do we modify clinical trial structures in order to get approval of drugs as quickly as possible?
Right, because the randomized trial was introduced in order to have be able to measure a detectable difference in survival with treatment of physician's choice versus
it's true, but you know, in the 80s, there was a epidemic, the HIV epidemic, which was every bit as dramatic as the COVID pandemic, maybe a little bit more, because every single person that got AIDS died, and at the time, it was a very sophisticated population. So a lot of it was particularly in the gay community, and a lot of the people in the gay community at the time who were speaking up were very sophisticated in a variety of communication and business venues, and they pushed back on the FDA, and they demanded an acceleration of the development. And they said to the FDA, why would you do a randomized study if everybody dies? Any drug that prevents someone from dying has to have efficacy. And the FDA was doing these randomized studies where half got a placebo and half got the test drugs. And by the way, one of the test drugs was discovered here. Mark Wainberg discovered 3TC here. And they actually said to the FDA, it was a small group called ACT UP, very heroic people, we're going to mess with your clinical trials. We're going to have pill parties. Everyone on the study is going to get together, put all their pills, placebo, and active agents in a pile and redistribute them. So we'll screw up your studies completely. They threatened them. And the FDA backed down and accepted accelerated pathways to drug approval. And so they probably shaved years off the development of active drugs that saved lives. And that's our goal here as well. And that's why maybe the most important component, and something that we're trying to include as well in MCTRC, is the voice of the patient. We need to have patient advocates and patients, former patients, because they have a very loud voice in the discussion in terms of moving these things forward.
Mm-hmm. And oftentimes they want access to a drug just because it might be their only chance. They don't want to be given a placebo.
Exactly.
But I do understand in certain cases the randomized trial is necessary because you can't replace a current therapy that we know works with something that might not work. And so at a late stage setting, we can try these combinations and see but if there's something that is coming up as being so effective, like you said, exactly. with 3TC, with it might have been the case for other drugs.
Well, it was easy.
We almost have no choice.
Yeah, exactly. So, for example, in melanoma, it has been one of those brick walls that we've hit for decades and decades. And, there was, in rapid succession, the recognition of a specific BRAF mutation and a BRAF inhibitor, and suddenly those patients had a dramatic response, but they started to develop other kinds of skin cancers, and there were other side effects. And very quickly after that, immune checkpoint inhibitors started to be tested, and they completely replaced the BRAF inhibitors within a couple of years. But these drugs, the drugs that target ALK, for example, in non-small cell lung cancer, these drugs were approved based on a couple of hundred patients in a phase 2 trial because there's nothing else that makes a tumor shrink than an effective therapy. So, ALK translocated non-small cell lung cancers are maybe 3% of lung cancer. So it's a rare disease. But the first person in the world treated with the first generation of ALK inhibitors was in Japan, and he was treated in an ICU, and he left on his own 2 feet a couple of weeks later because there's rapid tumor response. And now we're into a third generation of ALK inhibitors, but these things don't require randomized clinical trials. Nothing else but an effective therapy makes a tumor shrink. And if you don't have anything useful, hitting them with lots of toxic chemotherapy doesn't really, you're looking at the margins. So when we see a dramatic effect in a small number of patients, it actually has been enough to get approval for some drugs, and that's what we're pushing towards.
So it might slowly start to become more and more prevalent that we can accelerate this process. And yeah, especially with, like you said, all of these very, very effective and targeted drugs.
There are arguments now in the literature about progression-free survival being an endpoint and not having to wait for overall survival, because the wait, if you're waiting, you can calculate lives lost. And more often than not, progression-free survival is an indicator of an effective therapy. And if you're doing studies that have crossovers, the patients that progress, say in the standard arm or placebo arm, and then they get the experimental arm, that screws up your whole calculation of the overall survival. There are all kinds of confounding variables. So there's a very active debate in the literature now about the most efficient way to do this. Should regulators approve drugs just on the basis of progression-free survival, which may only take a year, and then wait to see if it's proven out with overall survival, and if not, well, then reconsider it? Or should they wait all of that time and have patients waiting who otherwise could have benefited from it? So we're looking at, and we're all pushing with patients, scientists, and clinicians, accelerated approval with additional data is what it's called, and that's where we're trying to go. And I've been really, really vocal about that in public fora, that patients can't wait, that we should be moving much more quickly. So it's not only the scientists in me, but it's the clinician in me that drives this kind of will.
Yeah, and I guess the patient advocacy is also critical in that space.
It is. I think, my heroes are the ACT UP people from the HIV epidemic, because they, first of all, their lives were on the line, so they didn't really care. They were desperate, but they changed. They forced regulators to change. They humanized the patient, and we have to ask ourselves in this day and age, when heaven and earth was moved because of the COVID pandemic. Huge expenditures and all kinds of systems changes, setting up screening clinics and vaccine clinics and all of that, clearing space in the hospital, delaying other treatments or other diagnostic tests, cancer screening and everything to treat COVID patients, all of which was necessary. But in its worst state, of COVID at the very darkest time, it was maybe 1/4 as many deaths due to COVID as due to cancer. So why aren't we in a perpetual state of alert and alarm with cancer? Why has it ever become acceptable that people die of cancer when there are potentially therapies out there?
Mm-hmm. A big part of it is communicating and getting these therapies and to treat the right patients.
And part of it is podcasts like this. See, students and scientists, clinicians can make a difference. They can both be very good scientists and also raise their voices and talk to patients and push. And I think we're by nature a little bit. We're all modest, and we're all a little bit loath to talk publicly about science and to get into that kind of advocacy, but I think that's something that we have to do. One of my models is the late Mark Wainberg, who was a virologist and helped screen one of the first active anti-HIV drugs But he understood that unless you get it to patients, it's really a nice discovery, but it's not meaningful. So when he was the president of the International AIDS Society, he brought it to Africa. He convinced pharma to make the drugs available to underserved countries, underdeveloped countries. And so the advocacy is part and parcel of the discovery, I think.
Interesting. So we need to get back to the war on cancer, I guess, which was a long time ago in the pre NCI, I think.
Yeah. We all sort of objected to the kind of militarization of the cancer research, but we can find some better words. It doesn't have to be a war, but maybe it's a grind. But we have to. The circle's bigger. Everybody's affected by cancer. Everyone has someone in their family who's experienced this, and so it touches all of us. And we should be able to reach that feeling in everyone, including the regulators. They consider themselves potential patients. Their family members have suffered from cancer as well. So we have to find a way of moving all of this forward, because fundamentally politicians, everybody, when you get down to it, they're humans concerned about the same thing. And that's what we have to find common cause. So that shouldn't distract us from the science and the translational research, but I think it's part of it as well. If we're thinking about ultimately making something that we can test in a patient, if it tests positive, maybe we have a responsibility as well to make sure that it actually gets to the patients.
Great. So now to end off the podcast, I have two questions. Mm-hmm. The first one being, how do you envision cancer therapy 5 to 10 years from now?
There's a couple of answers to that, but I love looking at the near and distant future, because I think it's entirely possible to see where we're going. I believe that within 5 to 10 years, for some tumor types sooner than for others, people will be dropping off either a tumor biopsy or a blood sample, because we're moving to liquid biopsies for diagnostics as well, at a central lab. And that a few weeks later, they'll be going by a central pharmacy to pick up, or maybe a drone will be delivering it to their front door, their personalized precision medicine package. Physicians will not be valued so much for their knowledge, because with AI, the robot will always know more than I will know. But we'll still have a role, maybe the role that physicians used to play a long time ago, reassuring the patients that the algorithm is correct, looking at side effects, talking to the family, reassuring and more of a supportive role. Maybe it'll be in a hologram in their living room rather than in a clinic. But I think that's where we're going. But even more than that, I think we're starting. We do all the as we start to look at the cancer statistics. Even this past year in Canada and the States, we see that although we're really doing dramatic benefits in terms of our treatments, cancer incidence continues to go up. And so our focus now has to really turn towards prevention. We know enough about genomics and family history and some other factors. For example, in breast cancer, if you combine breast density with family history with some genomic markers, you can find high-risk groups. And we shouldn't be recommending mammograms based on age. It should be based on something specific, precision screening. And I think that's where we're going to get to. We have a very low uptake of screening for colorectal cancer. People just aren't doing it. But if you go to some people and say, you know what, these tests that we've done tell us that your risk of colon cancer is higher than your neighbor's, this guy is going to get screened. He'll be highly motivated to get screened. So if in every CLSC, in every clinic, we had an iPad and people filled in their family history, and then someone took a buccal smear to get some DNA, and we could start to screen. Maybe maybe an anal swab to look at the microbiome, and we profiled the entire patient population, we would start to see subgroups that should be screened, and we'll find earlier cancers. Then the next step would be, some of these are lifestyle changes, but they're pharmacologic agents that could be preventive. Until now, it's been hard to convince pharma to do clinical trials of preventive agents, because what are you going to do? You're going to treat 1,000 people hoping to show that you've decreased the incidence of X, Y, or Z. It's a very complicated, expensive, and long-term study. But if you have a subgroup that has a higher risk, you can compare it to another group, and you can actually test a preventive agent. So if we start to profile our population, we will draw pharma into that space and start to prevent cancer. And I think that's the next horizon that will be extremely important as well.
And hopefully detection will become even more sensitive. And
yes.
These actually just doing genetic testing and I guess microbiome testing, whatever that looks like for the future, will just become more accessible and
it'll be more
feasible.
Exactly. And even all of the radiologic imaging, it'll be much more sensitive. Again, it'll be robot driven. As we're seeing for sort of mammogram exams, and even interactions with patients will be largely robot driven to get basic information, but much more efficient. Robots never get tired; they never are in a bad mood. They don't have to go to the washroom; they're steady. And so the introduction in a smart way of AI into reading radiology. And even sharing information is something that's going to be very much a part of the near future.
Yeah, I guess you can see it coming after watching things develop and evolve in the last number of years. Yeah,
there is a trajectory, and it's a trajectory that's accelerating in pace. But I mean, our discoveries in the lab are much faster. We're spending less time collecting specimens and doing analysis. Then rather than collecting high-quality data sets, which are already there. As we get more into a world of open access and data sharing, all of this is going to move much faster.
Great. So I just have one more question for you.
Sure.
This is for the students, because I think that a lot of our listeners will be students that are really interested in translational research.
I hope so, yeah.
Yeah, and so I just wanted to ask you for a piece of advice to students that are interested in getting into translational research, whether it be in school or in their career.
Yeah. First of all, I think, everyone's different, but I think it's very important to close as few doors as possible. Means make decisions that don't close doors, keep things open as long as possible. At some point you do focus, and that does close some doors. Find really good mentors. And by that, I mean a mentor that exhibits some element of selflessness. When I was doing my postdoc at the NIH, at one point my boss came to me and said, the next 2 papers, I'm taking my name off the paper. It's going to be yours so that you can succeed with grants. That's what you're looking for in a mentor, someone that is interested in your success. And colleagues, people that you can talk to and exchange ideas with. Be open, read widely. Even if you're focused on something very small, read widely, go to meetings. I think that's really, really important. And be curious and enthusiastic and work hard. All of the usual things. But I think in academia, it's very important to find the right mentor and the right environment.
I agree completely. I think that's great advice and a great place to leave the end of the podcast. So thanks again for joining us. It was a really nice discussion.
For me as well.
Yeah.
Thank you.
And I'm sure we'll be talking again.
Yes. Appreciate it.
Thank you for listening to the first episode in a series of many more to come. If you'd like to learn more about Dr. Batist and all the fantastic work that he's doing, as well as the MCTRC and the Tremplin Consortium, I have included some links in the description, so please check them out. Stay tuned also for our next episode, which will be hosted by another member of our committee, Myrta, and she will be discussing with Dr. André Diamant, CEO of Gray Oncology Solutions, and they will be talking about his journey from student to entrepreneur. It's going to be a really, really interesting episode, so keep an eye out on LinkedIn and Spotify in the next few weeks. And thanks for listening. See you next time.